long before bones turned to stone and before mountains rose where oceans once existed, vast forests covered much of the prehistoric world. These forests were not like modern forests. The air was warmer, richer in carbon dioxide, and heavy with humidity. Giant conifers towered over ferns taller than humans. Insects were abundant beyond imagination, filling the air with movement. In this ancient environment, trees produced enormous quantities of sticky resin as a natural defense against damage. Broken branches, fungal infections, and burrowing insects all triggered the flow of this thick golden liquid. Resin was not sap. Sap carried nutrients through a tree. Resin was protection. It oozed outward and hardened on contact with air, sealing wounds and trapping anything that tried to invade. When beetles chewed bark or ants tunneled into wood, they often found themselves stuck instantly. Wings froze mid-beat. Legs stretched outward in struggle. The more the creature moved, the deeper it sank. Within minutes, the surface hardened, locking the animal into a transparent prison. Over time, fresh flows of resin covered older, hardened layers. Storms knocked branches down, burying them beneath soil and sediment. Rivers carried the resin lumps into deltas and coastal plains. There they remained, sealed away from oxygen and bacteria. Millions of years passed. Pressure increased. Chemical reactions slowly transformed the resin into a stable substance. The volatile oils evaporated, and molecular bonds reorganized. The material hardened into amber. Unlike stone fossils formed from mineral replacement, amber preserved original biological material. Mussels did not rot away immediately. Microscopic structures remained intact. Even pigments sometimes survived. The organism inside was not flattened into an outline. It remained three-dimensional, as if frozen in time. Humans first encountered amber long before science existed. Along the shores of ancient seas, golden stones washed onto beaches after storms. They were warm to the touch and could burn with a pleasant smell. Early civilizations polished them into ornaments and amulets. They noticed tiny insects inside, but did not understand their origin. Many believed the creatures were born within the stone itself. Centuries later, natural philosophers began to suspect a different explanation. When heated amber released a pine-like scent, this hinted at a botanical origin. By the 17th century, scientists proposed that amber was hardened tree resin from an ancient forest. The trapped insects were not creatures born in stone, but victims of a prehistoric environment. The realization transformed amber from decoration into scientific treasure. It was not simply a gemstone, it was a biological archive. One of the earliest scientific examinations revealed flies whose wings still displayed delicate vein patterns. Under magnification, tiny hairs on their legs were visible. This level of preservation was unheard of in traditional fossils. In sedimentary rock, insects usually appeared as dark impressions. Here, they were complete organisms. Researchers began collecting amber from deposits around the world. The Baltic region produced large quantities dating to roughly 44 million years ago. In Myanmar, much older deposits dated to the age of dinosaurs, nearly 100 million years ago. The Dominican Republic revealed tropical ecosystems from a more recent era. Each deposit represented a different ancient forest, frozen in chemical time. As collections expanded, scientists noticed something remarkable. The amber pieces were not random. Many contained clusters of organisms together. Pollen grains floated beside beetles. Mites clung to feathers. Fungal spores surrounded plant fragments. Instead of isolated fossils, entire micro-ecosystems appeared preserved in a single drop of resin. This meant the resin flows captured living scenes, not dead remains. Creatures were trapped exactly where they lived. A mosquito hovering near a leaf remained near that leaf forever. A spider descending on silk thread stopped in mid-descent. Even droplets of water trapped within resin occasionally contained microorganisms. The implications were profound. Amber allowed scientists to study behavior, not just anatomy. It revealed interactions that bones could never show. Predation, parasitism, and pollination could all be observed directly. The ancient forest became visible again through countless small windows. One early examined specimen showed an ant carrying a larva in its jaws. The insect colony had been disturbed, possibly by the resin flow itself. The worker attempted rescue but failed. The entire scene hardened instantly. Another specimen preserved two mating flies connected at the abdomen. Their final moment lasted millions of years. The clarity of amber came from its chemistry. The resin excluded oxygen and contained natural antimicrobial compounds. Bacteria could not easily decompose trapped tissue. Dehydration occurred slowly, preventing collapse. As polymerization progressed, the surrounding matrix stabilized the specimen. Instead of decay, preservation occurred. Not every organism survived equally well. Larger animals rarely appeared because they escaped before sinking deeply. Small arthropods dominated the record. Yet, occasionally more complex remains appeared. Feathers, hair, and fragments of skin indicated larger animals had brushed against resin-coated surfaces. Even these traces were valuable, offering evidence of creatures otherwise unknown. By the 19th century, microscopes improved enough to study internal structures. Researchers observed compound eyes intact down to individual lenses. Some specimens revealed gut contents. Pollen grains inside digestive tracts indicated diet. Others showed parasites attached to hosts, proving ancient relationships. Amber became a time capsule, unlike any other fossil medium. Rock fossils recorded death after burial. Amber recorded life interrupted. Collectors and scientists often described opening a new specimen as witnessing a moment from deep time. The creature inside did not look ancient, it looked recent. Legs bent naturally, wings retained transparency. Some insects even reflected light as if alive. The mind struggled to accept the immense age separating observer and subject. The growing number of discoveries established amber as a primary source for reconstructing prehistoric forests. Climate conditions could be inferred from plant species. Insect diversity revealed ecological complexity. Certain pollinating species indicated the presence of flowering plants earlier than once believed. This understanding changed paleontology. Dinosaurs dominated land during the Cretaceous period, but amber showed that beneath their feet existed dense ecosystems of small organisms forming the foundation of food webs. Without them, larger animals could not survive. Amber therefore preserved the hidden majority of prehistoric life. Bones represented giants. Amber represented everything else. By the time modern analytical techniques emerged, scientists realized these golden stones held far more information than previously imagined. Chemical residues, microscopic air bubbles, and preserved pigments promised insights into ancient atmospheres and coloration. What once served as jewelry became one of the most detailed records of ancient biology ever discovered. The forest that produced the resin vanished long ago, yet its smallest inhabitants remained suspended in perfect stillness. The first chapter of amber research revealed not just preservation, but the existence of an entire lost world waiting to be examined piece by piece. As scientists continued examining amber deposits in greater detail, they began to notice that the resin did not flow randomly across the forest. Trees release resin in predictable situations. Storm damage, insect attacks, fungal infection, and sudden temperature stress all triggered heavy outpourings. Because of this, amber preserved moments of disturbance rather than ordinary calm conditions. Many of the trapped organisms were caught during events of chaos in the forest. In several Baltic specimens, researchers found dense swarms of tiny flies all trapped in a single layer. This suggested that a large resin flow occurred near a breeding ground. The insects likely gathered around decaying plant material when a sudden rupture in bark released a flood of sticky resin down the trunk. Those closest to the flow were immobilized immediately. Others landed on the hardened surface, unaware it was lethal, and became trapped as well. Layer upon layer accumulated within minutes. The speed of entrapment was critical to preservation. Resin hardened quickly on the outer surface, but remained soft underneath. A struggling insect could break through the thin crust and sink into the softer interior. This process prevented exposure to air and halted decomposition. Within hours, the animal was sealed beyond recovery. Within days, additional flows could bury the same specimen deeper, protecting it from sunlight and weather. Some amber pieces showed repeated flows separated by thin boundaries. Each boundary marked a different day or even a different season. When examined under magnification, scientists could reconstruct a timeline of events. A first layer might contain pollen carried by spring winds. A second layer held summer insects. A final layer trapped autumn spores. The amber recorded not only life, but also the passage of time in the forest. One remarkable discovery involved a small branch entirely encased in resin. On the bark were feeding beetles, fungal filaments, and mites living among them. The tree attempted to defend itself by releasing resin around the infected area. Instead of a single victim, the entire miniature habitat became entombed. The organisms remained exactly where they lived, preserving their spatial relationships. Scientists could map positions and understand how they interacted in life. Chemical analysis also revealed that resin composition varied between tree species. Some resins were thin and flowed quickly, capturing flying insects. Others were thick and trapped crawling animals near the trunk. This explained why certain deposits contained more ants while others contained more mosquitoes. The type of tree determined the type of fossil record. In tropical deposits, amber frequently included plant fragments such as petals and leaves. These were delicate structures, rarely preserved in rock. Their presence helped identify the ancient forest's vegetation. From these pieces, botanists reconstructed extinct plant lineages and traced the early spread of flowering plants across continents. The clarity of certain amber pieces allowed light to pass almost perfectly through them. Under strong illumination, tiny air bubbles could be seen surrounding the organisms. These bubbles formed from gases released by the trapped body as it dehydrated. The bubbles sometimes outlined soft tissues that would otherwise be invisible. In some cases, even individual muscle fibers could be inferred from their impressions. Scientists gradually realized that amber preservation captured a transition between life and death rather than the aftermath. In rock fossils, organisms were often transported by water before burial. Their position rarely reflected natural behavior. In amber, posture was genuine. A predatory insect still held its prey, a larva still clung to a leaf vein. Even the direction of movement was preserved. This accuracy allowed researchers to test theories about ancient ecosystems. For example, the abundance of pollinating insects in certain layers indicated that flowering plants were already diverse at that time. Previously, paleontologists believed such plants appeared later. Amber forced a revision of botanical history. The presence of juvenile insects also became important. Larvae and pupae stages were rarely fossilized in sedimentary rock because they decayed quickly. Amber preserved them intact. These developmental stages revealed life cycles, growth patterns, and metamorphosis processes of extinct species. Scientists could compare them to modern relatives and determine evolutionary relationships with much greater confidence. Occasionally, multiple individuals of the same species appeared together in a single piece. Ant workers surrounded a queen. Termites clustered around eggs. These were not random gatherings, but social groups. Amber therefore confirmed that complex social behavior existed millions of years earlier than previously documented. As imaging technology advanced, researchers began scanning amber using high-resolution methods capable of penetrating the surface without cutting it. Internal organs, wing joints, and even mouth parts hidden by opaque areas became visible. Each scan revealed new anatomical details. The organism inside the amber was no longer just a surface specimen, but a fully preserved body available for study. One particularly informative specimen contained a small lizard foot pressed against the resin surface. The animal had escaped, but the skin and scales remained. Microscopic pigment cells were still present. These pigment structures indicated coloration patterns. Instead of guessing prehistoric appearance from bones alone, scientists could directly infer color and texture. The importance of amber extended beyond paleontology. It also influenced evolutionary biology. Because amber preserved delicate species that rarely fossilized elsewhere, it filled gaps in the evolutionary record. Entire insect families, known only from amber, showed intermediate features linking ancient and modern forms. These transitions helped clarify how present-day biodiversity developed. In time, researchers understood that amber did not represent a single moment in Earth history, but many separate forests across millions of years. Each deposit was a different chapter. When combined, they formed a nearly continuous archive of small life evolving alongside changing climates and continents. The golden stones once worn as ornaments had become scientific documents written in biological detail. Every piece carried a story from a vanished forest, captured in the instant a drop of resin-sealed life away from decay. Chapter 1 concludes with the recognition that amber is not merely preservation, but direct observation of ancient living worlds, recorded exactly as they existed at the moment of entrapment. Among all fossil types, amber alone preserves motion. Not movement itself, but the interruption of movement. Every trapped organism was doing something at the exact instant the resin reached it. Because the resin hardened rapidly, there was no time for the body to settle into a natural resting position. The result is a record of behavior frozen in progress. One of the earliest examples studied closely showed a small fly landing on a resin-coated surface. Its front legs were extended downward, preparing for contact. The rear legs remained lifted, still in flight posture. The wings were angled backward in the braking position used during landing. The insect never completed the motion. The resin's surface tension gripped the feet and prevented takeoff. Within seconds, the wings stuck as well. The final pose revealed not death, but interruption. In another specimen, a beetle appeared halfway emerged from a burrow in bark. Only the front half of its body extended outward, while the abdomen remained inside the wood fragment, sealed within the amber. The resin had flowed over the opening, trapping the insect during its exit. This confirmed that certain beetles lived beneath bark in the prehistoric forest, just as their descendants do today. Perhaps the most dramatic scenes involve struggle. One piece from the Cretaceous period contained two ants locked together. Their mandibles were embedded in each other's limbs. The posture suggested territorial combat between members of rival colonies. Neither ant released its grip before immobilization. Even the tension of their legs pressing against the surface could be observed. Without amber, such behavior would be impossible to prove from fossils alone. Mating behavior appears frequently in amber. Flies connected abdomen to abdomen are common finds, but some cases are more complex. A pair of mating insects were discovered attached to a plant fragment, indicating they had chosen a specific location for reproduction. The position of their bodies showed the orientation used during copulation. This allowed scientists to compare reproductive mechanics across millions of years of evolution. Feeding behavior also left clear evidence. A small true bug was preserved with its mouthpart inserted into plant tissue. The feeding tube remained extended into the leaf vein. Nearby cells of the plant showed damage patterns identical to those caused by modern sap-feeding insects. This demonstrated that plant feeding strategies used today were already established in ancient ecosystems. One specimen captured a mosquito during the act of drinking. The proboscis penetrated the skin of a vertebrate fragment embedded in the same amber piece. Though the host animal was only represented small fragment, the position showed active feeding. The abdomen of the mosquito was swollen, indicating fluid intake at the moment of entrapment. This provided direct evidence of blood-feeding behavior far earlier than skeletal fossils alone could reveal. Spiders provided some of the most vivid action scenes. A juvenile spider descending on a silk thread became trapped when resin flowed down the trunk beneath it. The silk strand itself remained visible, stretching upward out of the amber. The spider's legs were extended as if preparing to land on prey or ground. The strand indicated that silk production mechanisms identical to modern spiders already existed at that time. Another specimen displayed a web structure partially preserved along with the spider. Several small flies were already stuck in the web before the resin engulfed the area. The spider had approached one victim and wrapped it in silk. Both predator and prey were captured together. This sequence showed multiple behavioral steps: web construction, prey capture, and immobilization, all preserved in one object. The direction in which insects faced also gave clues about light and environmental orientation. Many flying insects inside amber aligned toward the same side of the resin piece. This suggested they had been moving toward sunlight when the resin flow occurred. Such patterns helped reconstruct forest canopy conditions and the position of the tree when resin was released. Some of the most unusual discoveries involved cooperative behavior. In one amber fragment, several termites surrounded eggs clustered inside a cavity. Their bodies formed a protective ring around the developing young. This arrangement confirmed parental care strategies similar to those seen in modern termite colonies. Without amber, such social organization would remain speculative. Larval stages revealed feeding patterns rarely preserved elsewhere. A caterpillar appeared partially eaten by a parasitic larva emerging from its body. The parasite had begun exiting the host when resin sealed both organisms. The moment demonstrated parasitism in progress, rather than inferred from damage marks alone. The relationship between host and parasite could be studied anatomically. Even accidental behaviors were recorded. A winged insect collided with a resin droplet mid-flight. The impact angle bent the wings backward, suggesting high-speed contact. The head faced downward, and the legs were pulled inward as if reacting to sudden resistance. The preserved posture captured a fraction of a second normally invisible to science. These scenes collectively changed the interpretation of prehistoric life. Instead of imagining static organisms known only from bones or shells, scientists could observe daily activities: feeding, mating, fighting, caring for offspring, and escaping danger. Amber functioned as a biological photograph, taken millions of years before cameras existed. The importance of these discoveries extended to evolutionary theory. Behavioral traits often evolve slower than physical structures. By comparing behaviors preserved in amber to those of modern species, researchers confirmed continuity across vast timescales. Hunting techniques, defensive postures, and reproductive strategies persisted with little change. In some cases, extinct lineages displayed behaviors absent in modern descendants. Certain insects held wings in unusual resting positions not seen today. Others used mouth parts adapted for feeding on plant species no longer present. These differences suggested behavioral diversity once greater than today's ecosystems. The more amber was studied, the clearer it became that it documented life's activity rather than merely life's existence. Every specimen represented an interrupted moment in an ancient forest. Chapter 2 continues by examining even more dramatic encounters where survival attempts ended instantly under flowing resin. Some of the most revealing amber specimens are those that record not simple activity, but sudden crisis. Resin did not always trap solitary insects. Sometimes it flowed during moments of pursuit, escape, or violent interaction. In these cases, the preserved postures are tense and asymmetrical, showing effort rather than routine behavior. A well-known example contains a small cockroach running across bark when resin spread beneath its feet. The insect lifted its front legs high, attempting to pull free while the rear legs remained buried. The antennae were stretched forward, not relaxed. This posture matched the escape response of modern cockroaches exposed to sticky substances. The fossil demonstrated that rapid neural reflexes seen today already existed tens of millions of years ago. Another specimen showed a springtail caught at the very edge of a resin drop. Its body was curved sharply downward, and the forked appendage used for jumping was extended. The animal had triggered its escape mechanism but was immobilized before launch. The frozen posture revealed exactly how the jump would have begun, allowing scientists to study locomotion mechanics from a single preserved instant. In tropical amber deposits, numerous insects appear oriented in the same direction, legs pushing backward against the surface. Researchers interpret this as group escape behavior. A resin flow probably advanced across bark, and multiple animals attempted to outrun it upward. The front individuals were trapped first, while those behind climbed onto them, only to become stuck as well. The layered arrangement shows a brief collective response to danger. Predators also reacted to resin as they would to any environmental threat. Like prey animals frozen calmly, this specimen displayed active resistance, showing that entrapment was not always instantaneous. Some of the most dramatic scenes involve aerial encounters. In one piece, two flies collided while both attempted to avoid the same resin strand extending outward from a trunk. Their bodies were pressed together, wings tangled, suggesting mid-air contact. The resin strand hardened around them before separation. The specimen demonstrated that resin sometimes formed hanging threads capable of trapping flying insects above ground level. A rare discovery captured a dragonfly wingtip touching resin while the rest of the body remained outside. The insect escaped, but the detached wing portion stayed behind. The torn margin of the wing showed mechanical failure during escape. This kind of evidence helps researchers understand how strong insect wings were and how much force they could endure. Parasitic interactions often ended abruptly in amber. A parasitic wasp was found inserting its ovipositor into a beetle larva embedded in wood. The needle-like structure remained inside the host when resin engulfed both. The wasp had begun laying eggs inside the larva, but never completed the act. The fossil confirmed that internal parasitism occurred in that ecosystem and revealed the exact orientation used during attack. Another specimen showed a mite clinging to the leg of a larger insect. The host appeared to be grooming, with its mouthparts reaching toward the parasite. The behavior suggests parasite removal attempts, common in modern insects. Resin halted the process midway, preserving both the parasite and the defensive reaction of the host. Occasionally, multiple species interacted in a single crisis scene. One amber fragment preserved an ant carrying food while a predatory spider approached from above. At the same time, a resin flow spread over the surface. The ant's body leaned forward as if running. The spider extended its front legs in attack posture, and both were immobilized before contact. The scene revealed a food chain interrupted before completion. Environmental hazards also appear recorded. Tiny air bubbles inside some specimens align along one side of trapped organisms. This suggests resin flowed downward rapidly, dragging air with it. Several insects were positioned head upward, indicating they were climbing against the direction of flow. Their orientation helped determine how resin moved across the tree surface. In another case, a group of bark lice gathered around fungal growth on wood. When resin covered the area, some individuals remained feeding, while others attempted to disperse. The contrast between relaxed feeding postures and extended escape legs within the same piece showed that not all organisms detected the threat simultaneously. Sensory awareness varied among species. Some specimens even preserve evidence of sound production attempts. A cricket-like insect had its hind legs raised in the position used for stridulation, the rubbing motion that produces chirping sounds. The leg movement may have been part of mating or alarm signaling. The immobilized posture suggests the insect reacted to disturbance at the moment resin reached it. Juvenile animals often display different responses than adults. Larval insects typically curled or contracted rather than ran. In amber, they appear tightly bent, reflecting instinctive defensive behavior. Adults, by contrast, stretched outward in directional escape attempts. These differences helped scientists understand developmental changes in nervous system response. The study of these interrupted actions showed that amber functioned almost like a sequence of frames from a slow-motion recording. Each specimen captured a fraction of a second. When examined collectively, they revealed patterns of behavior and stability. Frequent resin flows implied trees experience constant damage from storms, temperature fluctuations, or heavy insect activity. The forest was dynamic and hazardous, not a static environment. By the end of these analyses, researchers recognized that amber provided more than anatomy and more than routine behavior. It documented reactions to immediate danger. The organisms inside were not simply living, they were responding. Chapter 3 will move further into direct confrontations where predators and prey were sealed together before the outcome was decided. If amber captures motion interrupted, then the most informative moments are those involving direct conflict. Predation is rarely preserved in ordinary fossils. Bones may show bite marks, but they do not reveal the exact sequence of events. Amber changes that completely. It preserves predator and prey in the same instant, allowing scientists to observe hunting behavior that once existed only as speculation. One of the most studied specimens contains a spider attacking a small fly. The spider's front legs are wrapped around the prey, while its fangs remain embedded in the thorax. The fly's wings are extended outward in a final attempt to escape. The resin flowed across the web at the exact moment of capture. The silk strands remain visible, connecting the predator to surrounding anchor points. This single piece confirmed that web-building spiders used trapping strategies nearly identical to those of modern species. In another case, a predatory beetle larva grasped a smaller insect with sickle-shaped jaws. The prey's body bent backward under pressure. The larva had already pierced the outer shell, but had not begun feeding. Because the resin hardened instantly, scientists could examine the feeding apparatus in action rather than at rest. The jaw's angle revealed how force was applied during attack, something skeletal remains could never show. Ants provide some of the most dramatic conflict scenes. A fragment from Burmese amber shows multiple ants swarming a termite worker. Several ants bite the legs, while another grips the abdomen. The termite's mandibles are open, possibly releasing defensive chemicals. The encounter resembles modern territorial raids, where ant colonies invade termite tunnels. This fossil demonstrates that organized group warfare among insects existed during the age of dinosaurs. Another specimen preserved an ant attacking a much larger insect pupa. The ant's body leaned forward while its abdomen curved beneath it in a stinging posture. The stinger penetrated the soft tissue of the pupa when resin sealed both animals. The behavior indicated active hunting rather than scavenging, showing that ants were already major predators in their ecosystems. Predatory flies also appear frequently. One amber piece contains a robber fly gripping another insect mid-flight. Its long legs hold the victim while the mouthparts pierce the body. The prey's wings remain extended backward, indicating it was captured in the air rather than on a surface. The capture posture matches modern aerial hunters that intercept targets while flying. Not all predators succeeded. A tiny parasitic wasp approached a caterpillar, its ovipositor extended toward the host. At the same moment, a drop of resin descended from above. The wasp became stuck before inserting eggs. The caterpillar remained untouched. This rare example shows an attempted attack interrupted before completion, offering evidence of predator avoidance by chance rather than defense. In several cases, multiple trophic levels appear together. A spider holds a fly, while a mite clings to the spider's leg. Resin sealed all three simultaneously. The mite likely fed on the spider's body, fluids or debris from prey. The arrangement revealed a miniature food chain inside a single drop of resin. Predator, prey, and parasite combined. Some confrontations were even more complex. One specimen includes a lacewing larva attacking a small aphid while ants stand nearby. Modern ants protect aphids for the sugary secretions they produce. The ants appear to be approaching the larva, possibly to defend their resource. The moment froze before intervention. This scene indicates cooperative behavior between ants and plant-feeding insects existed millions of years ago. Occasionally, predators themselves became prey. A small spider is preserved with a larger predatory wasp gripping its body. The wasp's mandibles hold the spider, while its stinger curves forward beneath the abdomen. Resin engulfed them before the paralyzing sting finished. This fossil provided direct evidence that certain wasps hunted spiders to supply food for their larvae. Even aquatic predation appears in rare amber pieces formed near water surfaces. One droplet trapped a small crustacean alongside a predatory larva adapted to shallow pools. The larva's mouthparts grasped the crustacean, while the surrounding resin included water bubbles, showing the event occurred at the boundary between liquid water and tree resin. The preservation of these encounters helped clarify ecological roles. Many species once classified as scavengers were revealed to be active hunters. Mouthparts, previously considered simple, were shown piercing prey. Behavioral assumptions, based on anatomy alone, were corrected by direct observation. Predation intensity could also be estimated. High frequency of attack scenes suggested dense populations and strong competition. The prehistoric forest was not peaceful but filled with constant interaction. Small animals faced danger as frequently as large animals did in later eras. Researchers also learned about defensive adaptations. Some insects displayed raised spines and expanded shields positioned between themselves and attackers. Others had chemical glands exposed during confrontation. Because the attacks were frozen mid-process, scientists could see how defense mechanisms were deployed rather than merely present. Amber therefore recorded ecological relationships with extraordinary clarity. Each predator and prey pair acted as proof of interaction, not inference. The forest ecosystem emerged as a network of direct connections rather than isolated species lists. The accumulation of these scenes established amber as the most reliable evidence for ancient food webs. Bones might show who existed, but amber showed who ate whom. Chapter 3 continues with even more specialized hunters whose methods were preserved down to microscopic detail. Beyond simple attacks, some amber specimens preserve highly specialized hunting strategies that depend on precision timing. These fossils reveal not only that predators existed, but exactly how they captured prey in ways rarely observed, even in modern ecosystems. One remarkable specimen contains a trap-jaw ant in the middle of a strike. Its mandibles are opened to an extreme angle, far wider than resting position. Modern trap-jaw ants close their jaws at extraordinary speed, snapping shut in less than a millisecond. The fossilized ant shows the moment just before closure. The prey insect stands directly between the jaws, unaware of the imminent impact. The resin sealed the scene before contact occurred, preserving the mechanics of a strike that normally leaves no trace. Another piece preserves a lacewing larva carrying debris on its back. These larvae disguise themselves by attaching plant fragments and dead insects onto specialized bristles. The larva approached a small, soft-bodied insect while camouflaged. The prey remained still, apparently not recognizing the predator. Both became trapped in resin before the attack completed. This confirmed camouflage-based hunting was already well developed in ancient forests. Some predators relied on chemical lures rather than speed. A spider, preserved in amber, displays droplets along its web threads. Nearby flies appear positioned toward those droplets rather than randomly entangled. Modern spiders sometimes produce attractive secretions to mimic food sources. The positioning of the flies suggests they approached voluntarily before becoming trapped. Resin captured the web, droplets and victims simultaneously, providing rare evidence of deceptive hunting. A different specimen contains a predatory larva extending a tubular mouthpart toward a mosquito larva. The structure resembles a suction device used by certain modern aquatic insects. The prey's body shows compression as fluid extraction begins. This suggests fluid feeding predation methods evolved very early and remained stable across millions of years. One of the rarest scenes shows a group hunting strategy. Several ants surround a large, struggling insect, while one individual grips the antenna and another bites the joint of a leg. The arrangement indicates coordinated roles rather than random biting. Resin froze the positions before the prey collapsed. Cooperative predation among insects, therefore, predates many modern animal group hunting behaviors. Predators also adapted to specific environments within the same forest. Bark-dwelling hunters appear flattened with wide legs spread outward, pressing prey against surfaces. Flying hunters, in contrast, have long, narrow limbs designed to intercept targets in open air. Amber preserves both strategies side by side, demonstrating ecological specialization within a small area. Some prey animals possessed defensive countermeasures. A beetle is preserved, releasing a cloud of microscopic droplets from its abdomen toward an attacking ant. The droplets remain suspended in the amber around the two insects. Modern beetles use chemical sprays to deter attackers. The fossil proves such chemical defense systems were already active in that prehistoric ecosystem. There are cases where both predator and prey became victims simultaneously. A spider captured a fly in its web. Then a resin flow covered the web. The spider appears to have attempted escape, abandoning the prey and moving toward the edge of the resin. Its legs stretch outward in a climbing motion, while the prey remains wrapped behind it. The sequence shows decision-making behavior, prioritizing survival over food when threatened. One specimen preserved a mantis nymph grasping a butterfly-like insect. The mantis forelegs held the prey while the head rotated sideways, possibly reacting to the resin touching its abdomen. The prey's wings were partially folded, suggesting it had just landed before attack. The combined posture indicates a classic ambush strike from a concealed position. Tiny parasites also acted as hunters on a smaller scale. Mites attached themselves near joints of larger insects where protective armor was thin. Some are preserved with mouthparts inserted into soft tissue. The host's leg bends inward, possibly in reaction to irritation. Such fossils reveal parasitic feeding behavior not detectable from isolated remains. The variety of hunting techniques preserved in amber shows evolutionary experimentation. Speed-based capture, camouflage, chemical deception, coordinated attacks, and fluid extraction all existed simultaneously. The forest supported a complex network of survival strategies. These scenes also demonstrate that many ecological roles have remained stable over immense timescales. Modern insects still use nearly identical techniques. Evolution refined details, but rarely replaced successful strategies entirely. Amber, therefore, connects present ecosystems directly to ancient ones through behavior rather than only anatomy. As the number of specimens increased, scientists began reconstructing full food chains. Plant-feeding insects supported predators, which in turn hosted parasites. Each interaction appears somewhere within amber collections. By combining thousands of individual moments, researchers assembled a continuous picture of life interacting under constant pressure. The study of predator and prey in amber ultimately transformed understanding of prehistoric environments. The ancient forest was active, competitive, and densely populated. Every surface supported life, and every life form depended on another. The resin that once protected trees inadvertently recorded the entire struggle for survival occurring beneath their branches. Chapter 4 will move beyond insects to examine a discovery once considered impossible: traces of feathered creatures and small vertebrates preserved inside amber itself. For many years, amber was considered a medium limited to small organisms. Insects, mites, and plant fragments dominated collections, while larger animals seemed absent. The assumption appeared reasonable. A large creature would struggle free before sinking deeply into resin. Yet, scattered discoveries began challenging that belief. Instead of entire bodies, fragments appeared. Feathers, hair, and pieces of skin embedded in perfectly clear amber. One early specimen revealed a simple filamentous feather, unlike modern bird feathers. Under magnification, it showed no central shaft and branched loosely along its length. This matched predictions of primitive feather evolution proposed from skeletal fossils. The structure suggested insulation rather than flight. Because the feather was preserved three-dimensionally, scientists could study the arrangement of barbs without distortion. This was the first direct evidence that some dinosaurs possessed soft coverings long before birds dominated the sky. Soon after, additional feathers appeared in older deposits dating to the mid-Cretaceous period. Some were more complex, with a defined central rachis and symmetrical veins. The veins indicated aerodynamic potential, though not necessarily strong flight ability. Pigment-containing cells were visible along the feather surface. By analyzing microscopic pigment structures, researchers inferred coloration patterns such as dark browns and reddish tones. Instead of guessing dinosaur appearance, They could now reconstruct realistic visual features. The feathers spread outward from all sides, forming a plume rather than a flat fan. This indicated the animal likely used the tail for display or balance rather than flight steering. The preservation was so complete that keratin sheaths around the feathers remained visible. Hair fragments also appeared in certain deposits. Unlike feathers, hair rarely fossilizes in rock because it decays quickly. In amber, individual strands remained intact with cuticle patterns preserved. Some hair was smooth, while other strands were barbed, indicating different mammalian species. These finds confirmed that small mammals lived alongside dinosaurs in the same forests. The most surprising discovery came when a lizard foot was found fully encased. Each toe displayed scales, claws, and soft tissue contours. The position of the foot suggested the animal stepped onto resin and escaped, leaving the extremity behind. Microscopic examination revealed pigment distribution across the scales, indicating striped coloration. Such details could never be recovered from skeletal remains alone. Additional specimens included portions of skin with scale patterns belonging to small reptiles. Some scales overlapped tightly like armor, while others were separated by flexible skin, suggesting different locomotion styles. The diversity implied a complex community of vertebrates inhabiting the forest canopy and bark surfaces. These discoveries expanded the scope of amber research dramatically. No longer limited to arthropods, amber began documenting vertebrate appearance directly. Paleontologists could finally examine surface features such as texture and color, rather than only bone structure. One piece preserved a tiny hatchling bird wing. The bones were fragile and partly hollow, indicating limited flight capability. The feathers were soft and downy, suggested by time. The molecules confirmed the biological origin of feathers and hair structures. This helped distinguish true biological preservation from mineral imitation. These findings also clarified evolutionary relationships. Some feather structures matched those predicted for non-flying dinosaurs, while others resembled modern birds. The combination supported the theory that birds evolved gradually from feathered dinosaur ancestors. Amber offered physical proof in addition to skeletal comparisons. Environmental interpretation also benefited. The presence of climbing lizards and perching birds suggested a dense forest canopy rather than open plains. The animals preserved in amber were those interacting directly with tree surfaces, reinforcing the idea that resin-producing trees formed a complex vertical habitat. Each fragment told part of a larger story. A feather drifting onto resin recorded molting. A claw mark embedded in surface layers showed attempted escape. Skin impressions preserved pressure patterns from movement. These details transformed extinct creatures from abstract skeletons into living animals with texture and color. By preserving delicate vertebrate tissues, amber bridged a gap between paleontology and biology. The organisms no longer appeared distant and alien. They appeared familiar, connected to modern ecosystems through visible traits. Chapter 4 continues by examining how these preserved features revealed not just appearance, but also behavior and ecological roles of feathered creatures living in prehistoric forests. Once fragments of feathers and small vertebrates were confirmed inside amber, scientists began focusing not only on appearance, but on how these animals behaved in their environment. The preserved tissues often contained clues about movement, growth, and daily activity. Instead of static anatomy, researchers could reconstruct moments from the lives of animals that lived alongside dinosaurs. One feather cluster, preserved in Burmese amber, showed irregular wear along the tips. The edges were frayed rather than smooth. This type of damage matches contact with branches during repeated movement through dense vegetation. The animal likely moved frequently among foliage rather than gliding in open air. The feather, therefore, indicated habitat preference, not merely structure. Another specimen revealed tiny parasites attached to a feather shaft. Several mites clung near the base where cleaning would be difficult. Their position suggested the host animal groomed itself regularly but could not reach certain areas. This provided indirect evidence of preening behavior in early feathered species. Grooming, a behavior common in modern birds, therefore existed in their ancient relatives. In a separate amber piece, a small bird hatchling wing displayed partially developed feathers, still enclosed in thin sheaths. These sheaths normally split as feathers mature. The intact condition implied the individual had not yet completed growth. Because the bones were lightweight and unfused, researchers concluded the animal had limited flight ability and probably remained in the nest. The presence of tree resin near the wing indicated the nest was built close to the trunk of a resin-producing tree. Some tail feathers contained alternating light and dark pigment bands. In modern animals, banding often serves camouflage by breaking body outline in dappled light. The pattern suggests these early feathered animals lived in shaded forest environments where concealment from predators was necessary. Amber therefore revealed ecological pressures acting on appearance. One remarkable specimen preserved the skin of a small reptile with a clear bite mark. The surrounding tissue showed compression patterns matching tooth spacing of a small predator. The reptile escaped, leaving damaged skin behind. This confirmed that predators attempted attacks on these animals within the same forest, and that survival sometimes depended on shedding or tearing free from a grasp. A lizard tail fragment provided additional behavioral evidence. Many modern lizards can detach their tails to escape predators. The preserved tail showed a clean separation plane consistent with self-detachment rather than accidental tearing. The animal likely sacrificed its tail when stuck in resin, escaping while leaving the tail behind. This indicates defensive strategies identical to those used by present-day reptiles. Several feathers were discovered arranged in loose piles within single amber pieces. The grouping suggested molting sites where animals shed old feathers periodically. Because multiple individuals were not present, the feathers likely accumulated beneath perches. Resin then flowed over the ground surface and sealed them together. This allowed scientists to infer seasonal molting cycles in ancient species. Color patterns derived from pigment structures also implied display behavior. Some feathers contained iridescent microstructures capable of reflecting light at changing angles. Such features serve no flight function, but are used in visual signaling, typically during courtship or territorial display. The existence of these structures indicated that visual communication already played a role in social interaction. Evidence of diet appeared as well. In one amber specimen, a droplet containing stomach contents was preserved near a small bird skeleton fragment. Inside the droplet were fragments of insect exoskeletons and pollen grains. The combination suggested the animal fed both on insects and plant material, indicating an omnivorous diet similar to many modern forest birds. The positioning of certain claws also provided behavioral clues. A claw fragment embedded in resin showed curvature adapted for gripping vertical bark rather than flat ground. The animal likely climbed tree trunks, hunting insects or moving between branches. This supported the idea that early birds and their relatives occupied diverse ecological niches within the forest. Perhaps the most revealing specimen showed multiple feather types attached to the same skin fragment. Long, stiff feathers lay beside shorter, insulating ones. This combination indicates layered plumage used for both temperature control and display. Such complexity implies advanced regulation of body heat and possibly active metabolism, bridging characteristics between reptiles and birds. These discoveries collectively transformed understanding of prehistoric vertebrates. Instead of generalized creatures inferred from skeletons, they became animals with habits, behaviors, and survival strategies. They groomed, molted, displayed, escaped predators, and raised young within forest habitats filled with resin-producing trees. Amber provided a direct record of these behaviors because it captured contact points between animals and their environment. A feather caught during grooming or a tail lost during escape preserved not just form but action. Each fragment represented interaction between living organism and surroundings. By the end of these studies, scientists recognized that amber was not merely documenting insects around dinosaurs, but entire living communities, including small vertebrates. The forest ecosystem appeared dynamic and complex, filled with creatures interacting constantly at every scale. Chapter 5 will turn attention to a different kind of evidence preserved in amber: blood-feeding insects and the microscopic traces they carried from their hosts. Among the most discussed discoveries in amber are blood-feeding insects. Unlike predators that leave visible bite marks on bone, these small organisms interact with soft tissue that rarely fossilizes. Amber preserves them at the exact moment of feeding, sometimes with traces of their last meal still inside their bodies. This provides rare insight into the relationships between parasites and hosts in prehistoric ecosystems. One specimen contains a mosquito with its abdomen enlarged far beyond its normal resting size. The proboscis remains extended forward, suggesting the insect had just withdrawn it from a host. Under magnification, dark material fills the abdomen. Chemical analysis showed iron-rich compounds consistent with digested blood. This indicated the mosquito fed shortly before becoming trapped in resin. Nearby, in the same amber piece, lies a fragment of feathered skin. The spatial proximity suggests the mosquito had been feeding on a small feathered animal when the resin flowed. Though the host is incomplete, the association demonstrates that blood-feeding insects interacted with feathered vertebrates long before modern birds diversified widely. Ticks are also preserved in remarkable detail. One amber tick is swollen with a partially transparent abdomen showing layered internal contents. The mouthparts are embedded in a piece of reptilian skin preserved beside it. The tick had attached firmly and was feeding for an extended period when resin sealed both organisms together. The positioning of the barbed mouthparts matches attachment mechanisms seen in modern ticks, indicating this method evolved very early. Fleas appear less frequently but provide equally valuable information. A fossil flea shows elongated hind legs for jumping and a flattened body suited for moving through hair or feathers. Its body orientation suggests it was clinging to a host surface when trapped. Because fleas cannot feed long without a host, their presence near hair fragments strongly implies parasitic behavior similar to present-day species. Some specimens preserve biting midges, tiny flies capable of piercing skin. Their mouthparts are thin and needle-like. In several pieces, these insects appear in clusters, suggesting swarm feeding behavior. Modern midges often attack hosts in groups. Amber, therefore, records not only individual feeding, but also collective parasitic strategies. Internal examination techniques have revealed microscopic particles inside these insects. Pollen grains sometimes appear alongside blood residues. This suggests that the insects alternated between feeding on plant fluids and animal blood, similar to modern species that require sugar meals between blood feedings. The dual diet indicates complex ecological roles. Parasites themselves were often parasitized. Mites attached to ticks are preserved in certain specimens, likely feeding on the tick's body fluids. This layered parasitism demonstrates a hierarchy of dependence, with each organism relying on another. Amber captures these relationships simultaneously, allowing reconstruction of multi-level interactions. Scientists hoped these preserved blood meals might contain intact genetic material from hosts. Early studies attempted to extract biological molecules from inside the insects. While fragments of complex organic compounds were detected, they were heavily altered by time and chemical processes. Complete genetic sequences could not be recovered. Nevertheless, the chemical traces confirmed the original biological nature of the contents. Some insects appear to have been interrupted during feeding. A mosquito with its proboscis still inserted into skin remains attached in amber. The abdomen is only partially filled, showing the feeding process had not finished. This demonstrates how quickly resin could immobilize organisms, capturing a moment lasting only seconds. One unusual specimen contains a louse clinging to a feather barb. Its specialized claws grasp the filament tightly. Lice feed on skin debris and sometimes blood. The presence of the louse attached to feather structure proves close physical contact with the host, offering evidence of grooming behavior necessary to control such parasites. Certain ticks preserved in amber show growth stages from tiny larval forms to larger nymphs. These stages require multiple hosts across their life cycle. The variety suggests diverse host species lived within the forest. Even without finding every host fossil, the parasite record reveals host diversity indirectly. These discoveries also provide environmental information. Blood-feeding insects require warm conditions and available vertebrate hosts. Their abundance in particular deposits indicates a thriving ecosystem rich in small animals. Regions lacking such insects likely represented cooler or less populated environments. Amber therefore records disease vectors as well as parasites. While actual ancient diseases rarely survive chemically, the presence of these insects implies potential transmission of pathogens among prehistoric animals. The forest was not only a place of predation, but also one of infection and immune response. By studying the anatomy of these insects, scientists traced the evolution of feeding apparatuses. The piercing mouthparts, anticoagulant saliva channels, and sensory organs for locating hosts were already specialized millions of years ago. This suggests parasitic feeding developed early and remained stable because of its effectiveness. The preserved insects reveal that prehistoric animals lived under constant pressure from parasites, just as modern animals do. Blood feeding, grooming, immune defense, and parasite avoidance were essential parts of daily life. Chapter 5 continues by examining how these preserved parasites helped reconstruct ancient host animals and the limitations of recovering biological information from such deep time remains. As the study of blood-feeding insects advanced, scientists realized that these parasites provided indirect evidence about animals that rarely fossilized. Large skeletons might preserve the existence of certain species, but many small vertebrates left almost no bone record. Parasites, however, required hosts to survive. By identifying what kinds of parasites existed, researchers could infer the presence of host populations even when no bones were found nearby. Different parasites specialize in different hosts. Some ticks prefer reptiles, others mammals, and others birds. The mouthpart structure often reveals which type of skin they are adapted to penetrate. When paleontologists examined the preserved ticks in amber, they discovered several varieties with features suited for gripping feathers. This indicated that feathered animals were abundant in that forest environment. The parasites confirmed what scattered feather fragments had already suggested. Small feathered vertebrates were common inhabitants. Similarly, certain fleas possessed comb-like spines positioned to anchor within fur rather than scales. This pointed to the presence of furry mammals sharing the same habitat. Because mammal bones from that period were rare and fragile, the flea fossils served as stronger evidence of mammalian diversity than skeletal remains alone. Some biting flies showed sensory organs tuned to body heat detection. Such adaptations are used by modern species to locate warm-blooded animals. Their existence in ancient amber suggests that warm-blooded hosts were present and widespread. This information helps refine understanding of metabolic evolution among prehistoric vertebrates. Researchers also analyzed the physical condition of parasites to understand feeding frequency. Some mosquitoes contained only partial meals, while others were completely engorged. This variation indicated multiple feeding opportunities within a short time span, implying host animals lived in dense populations rather than being scattered sparsely through the forest. Chemical examination of internal residues offered additional clues. Although intact genetic material was not preserved, fragments of complex organic molecules associated with hemoglobin breakdown were detected. The molecular ratios varied between specimens, implying they fed on more than one type of host blood. Even without identifying exact species, scientists concluded that multiple vertebrate groups coexisted in the ecosystem. Attempts to recover complete biological sequences faced serious limitations. Over millions of years, heat, pressure, and natural radiation break down molecular structures. Bonds fragment and rearrange into simpler compounds. While amber slows decay dramatically compared to open environments, it does not halt chemical change entirely. As a result, the preserved insects contain altered remnants rather than living biological material. Microscopic imaging techniques, however, revealed structural preservation beyond chemistry. Cells inside the insect abdomen still showed outlines of membranes. These were empty shells rather than functioning tissue, yet they retained shape. By studying these shapes, scientists confirmed that digestion had begun before entrapment. The parasites were active feeders rather than individuals that died long after feeding. Some specimens even preserved gut bacteria, shapes as faint impressions. Though the bacteria themselves were no longer viable, their presence indicated symbiotic microorganisms existed within parasites just as they do today. This suggested ancient ecosystems included complex microscopic communities essential for digestion and survival. Parasites also recorded seasonal patterns. Groups of similar insects trapped within the same resin layers implied synchronized emergence periods. Many modern parasites hatch during warm seasons when hosts are most active. The clustering of fossil parasites, therefore, hinted at climatic cycles within the prehistoric forest. In rare cases, parasites appeared, attached to hosts suffering injury. One tick remained anchored near a wound in reptile skin, preserved in the same amber piece. The wound edges were swollen, suggesting inflammation. This shows that immune responses existed and that parasites exploited weakened tissue areas, much like modern behavior. Another specimen preserved a feather fragment, heavily damaged along its shaft with several lice attached. The feather structure indicated repeated stress and wear, possibly from grooming attempts. The presence of multiple parasites implied prolonged infestation rather than brief contact. These findings reshaped the perception of prehistoric life. Ancient animals were not isolated giants wandering empty landscapes. They lived with constant biological pressures, biting insects, attached parasites, infections, and grooming needs. The ecological web extended down to microscopic levels. By comparing parasite diversity across different amber deposits, scientists could also identify environmental differences. Regions with abundant ticks and fleas likely supported many terrestrial vertebrates, while deposits dominated by biting midges suggested humid conditions near water sources. Thus, parasites became environmental indicators. Amber, therefore, functioned as an indirect census of hidden animals. Even when bones were absent, parasites testified that hosts once existed there. This approach broadened paleontology beyond visible remains into ecological reconstruction. The limits of molecular preservation reminded researchers that amber is a window into structure and interaction rather than a source of living material. Its value lies in understanding life processes, not recreating extinct organisms. The preserved parasites demonstrate relationships, behavior, and environmental conditions with clarity unmatched by any other fossil type. Chapter 6 will explore another dimension of this preserved world: plants, pollen, and entire microscopic landscapes captured alongside animals in the same drops of resin. While animals often draw the most attention, a large portion of amber's scientific value comes from the plant material trapped alongside them. Resin originates from trees, and as it flows, it collects microscopic particles from the surrounding air and surfaces. These particles include pollen grains, spores, fragments of leaves, and fungal threads. Together, they form a record of the ancient forest environment at a level of detail impossible to achieve through ordinary plant fossils. Pollen grains are especially informative. Each plant species produces pollen with a distinct shape and surface pattern. Under magnification, the outer wall of a pollen grain shows ridges, pores, or spikes unique to that species. When pollen becomes embedded in resin, it is protected from decay and distortion. Scientists can therefore identify the types of plants that lived in the forest even when no leaves or wood are preserved nearby. In one amber deposit, thousands of pollen grains were found surrounding a single trapped insect. The grains belonged to several different flowering plants, indicating the forest contained diverse vegetation. Because pollen can travel by wind or by insect transport, its location relative to animals reveals pollination methods. Pollen clustered on the legs and mouthparts of certain insects confirmed that those species acted as pollinators millions of years ago. Flower fragments occasionally appear preserved with intact petals and reproductive structures. These delicate tissues normally decay rapidly in open air. In amber, they retain their three-dimensional form. One specimen contains a complete miniature flower attached to a short stem, along with a small beetle positioned among the stamens. The beetle's body carried pollen grains matching the flower, demonstrating a pollination interaction captured at the moment it occurred. Fungal life is equally well represented. Fine branching filaments, known as hyphae, often grow across bark surfaces where resin flows originate. When trapped, they form intricate networks visible under magnification. Some hyphae penetrate plant tissue fragments, indicating decomposition processes already underway before the resin sealed them. This shows that decay organisms played a role in nutrient recycling in the ancient forest, just as they do today. Spores from fungi appear in enormous numbers within certain layers of amber. Their concentration can signal seasonal conditions. Modern forests release large quantities of spores during humid periods. Similar patterns in amber suggest cycles of wet and dry seasons existed in prehistoric climates. Plant fragments also include pieces of bark, needles, and leaf cuticles. The outer surface of leaves carries microscopic pores used for gas exchange. The density and size of these pores vary depending on atmospheric carbon dioxide levels. By measuring these structures in amber preserved leaves, scientists estimated the composition of ancient air. Higher pore density indicated lower carbon dioxide concentrations, while fewer pores suggested higher concentrations. Some amber pieces contain tiny droplets of water trapped alongside plant debris. Within these droplets are microscopic algae and bacteria. These microorganisms form the base of aquatic food chains. Their presence implies small pools or moist surfaces existed near resin-producing trees. The forest was not uniform, but included varied microhabitats. Lichens, symbiotic organisms formed by fungi and algae together, also appear preserved on bark fragments. Lichens grow slowly and are sensitive to air quality and humidity. Their occurrence suggests stable environmental conditions over long periods. The type of lichen indicates whether the forest canopy was dense or open to sunlight. Plant resins occasionally captured seeds and fruits in early developmental stages. Some seeds show adaptations for wind dispersal, such as thin wings, while others are smooth and compact, suited for animal transport. These differences reveal how plants spread across the landscape, and which animals likely assisted in dispersal. The association of plant and animal remains in the same amber pieces allows reconstruction of entire ecological relationships. An insect carrying pollen next to a flower fragment indicates pollination. A leaf with feeding damage beside a herbivorous larva shows plant consumption. Fungal hyphae invading a dead insect demonstrates decomposition. Each specimen acts as a miniature ecosystem rather than a single organism. By comparing plant types across different deposits, researchers tracked how vegetation changed over millions of years. Some ancient plant families present in amber no longer exist today, while others remain common. This helps map evolutionary transitions in global flora. Amber therefore preserves the foundation of the food web. Plants capture energy, herbivores feed on plants, predators feed on herbivores, and decomposers recycle nutrients. All these stages appear together within the same transparent medium. The study of plant inclusions transformed amber research from zoology into full ecosystem reconstruction. Instead of isolated life forms, scientists now viewed a functioning forest community frozen in place. Chapter 6 continues by examining how microscopic plant and fungal evidence reveals ancient climates and environmental changes across geological time. As more plant material from amber was examined, scientists realized that these preserved fragments could be used as environmental instruments. The forest itself left measurable signals in microscopic structures. By studying them carefully, researchers reconstructed temperature, humidity, and atmospheric composition from millions of years ago. One important indicator came from leaf cuticles. The outer surface of leaves contains tiny openings called stomata, which regulate gas exchange. Plants adjust the number of stomata depending on carbon dioxide concentration in the air. When carbon dioxide levels are high, fewer openings are required. When levels are low, plants produce more to capture sufficient gas. Amber preserved leaf fragments with stomata intact, allowing accurate counting. Measurements from multiple deposits revealed that certain prehistoric forests grew under significantly higher carbon dioxide levels than today. Temperature conditions could also be inferred from plant anatomy. Some conifer needles preserved in amber showed thick wax coatings. Modern plants develop similar coatings in warm environments to reduce water loss. Their presence suggested that the forest experienced high temperatures and strong sunlight at certain times of year. In contrast, other leaves displayed thinner surfaces and larger pores, indicating shaded or humid conditions beneath a dense canopy. Pollen provided additional seasonal information. Many plant species release pollen during short reproductive periods. When amber layers contained large concentrations of identical pollen types, researchers concluded the resin flowed during flowering season. Adjacent layers with different pollen assemblages indicated later seasonal events. By analyzing repeated patterns across many samples, scientists reconstructed annual cycles of plant growth. Fungal spores also revealed moisture levels. Certain fungi thrive only in damp conditions, while others prefer drier surfaces. When spores from moisture-loving species dominated a layer, the forest likely experienced heavy rainfall or persistent humidity. Alternating layers of dry-adapted and wet-adapted spores indicated changing climate patterns over time, rather than a constant environment. Some amber pieces contained trapped air bubbles large enough to analyze chemically. The gases inside were altered but still reflected proportions of ancient atmospheric components. Combined with plant pore data, these bubbles strengthened estimates of prehistoric air composition. The forest atmosphere was warmer and richer in greenhouse gases than modern temperate regions. Evidence of wildfire appeared in rare specimens. Tiny charcoal particles embedded alongside pollen indicated that fires occurred in nearby areas. After such events, resin production often increased as trees attempted to seal damage. Layers rich in charcoal and resin therefore marked ecological disturbance, followed by recovery. This showed that ancient forests experienced cycles of destruction and regrowth, much like modern ones. Water conditions were reconstructed through microorganisms. Some amber droplets enclosed freshwater algae species that survive only in shallow standing water. Their presence implied ponds or slow-moving streams near resin producing trees. Other droplets contained salt-tolerant organisms, suggesting coastal proximity for certain deposits. Thus, amber distinguished inland forests from coastal ones. Plant growth patterns also changed across geological time. Earlier, deposits contained mostly gymnosperm pollen from cone-bearing trees. Later, layers showed increasing diversity of flowering plant pollen. This transition marked the global expansion of flowering plants and the ecological shift that followed. Insects adapted to new nectar sources, and pollination networks became more complex. By mapping these botanical changes, scientists traced climate evolution. Periods with abundant tropical plant indicators corresponded to warmer global conditions. Later, deposits with temperate plant types reflected cooling climates. Amber, therefore, recorded not only a single forest, but long-term planetary change. These environmental reconstructions influenced understanding of animal evolution as well. The spread of flowering plants created new feeding opportunities, encouraging diversification of pollinating insects and small vertebrates. Climate fluctuations affected migration and extinction patterns. The biological community preserved in amber was shaped directly by its environment. Researchers eventually assembled regional climate histories using thousands of tiny inclusions. Each fragment contributed a small data point. Together they formed a continuous environmental record spanning millions of years, far more detailed than sediment layers alone could provide. Amber proved capable of preserving microscopic ecological evidence with extraordinary fidelity. Plant structures, spores, and trapped gases collectively described the atmosphere, weather cycles, and disturbances of ancient landscapes. Through these clues, scientists transformed isolated specimens into a living environmental narrative. Chapter 7 will examine some of the strangest organisms found in amber, creatures whose forms differ so greatly from modern life that they reveal entire evolutionary experiments that no longer exist today. Among the many organisms preserved in amber, some do not resemble any living species. They are not simply extinct relatives, but forms with body structures that appear experimental, as if evolution once explored pathways that were later abandoned. Because amber preserves small animals in complete detail, these unusual creatures can be studied far more accurately than fragmented fossils in rock. One insect discovered in Cretaceous amber possessed an elongated neck nearly as long as its entire body. Its head carried delicate jaws suited for piercing, while its wings resembled those of lacewings. The neck flexibility suggested it could probe into narrow spaces, such as bark crevices or plant cavities, to reach hidden prey. No modern insect combines these features. The species represents a feeding strategy that vanished, possibly because competing predators evolved more efficient methods. Another specimen revealed a fly with paddle-shaped antennae covered in fine sensory hairs. The exaggerated antennae likely detected faint air vibrations or chemical signals. Their size, however, would have hindered fast flight. The species may have depended on quiet environments where detection mattered more than speed. Changes in forest structure or predator pressure could have made such specialization disadvantageous, leading to extinction. Some amber insects display mouthparts combining traits of chewing and sucking feeders. These hybrid structures suggest transitional diets, perhaps feeding on both plant tissue and fluids. Modern insects typically specialize in one feeding mode. The mixed design may have been less efficient once more specialized competitors appeared. Larval forms are especially unusual. One larva, preserved in amber, carried long lateral extensions along its body, resembling thin spines but flexible. These structures may have increased surface area to sense vibrations or to deter predators. No comparable modern larva uses this exact adaptation. It indicates that ancient ecosystems hosted defensive strategies later replaced by chemical defenses or camouflage. There are also wing structures never seen today. Some insects possess wings with extra branching veins, forming a mesh-like pattern far denser than in modern species. The heavy vein network suggests slow, gliding movement in dense air beneath forest canopies. As atmospheric conditions and plant distribution changed, such wings may have become inefficient compared to lighter, faster designs. One group of extinct ants found in amber had upward-facing jaws rather than forward-facing ones. Instead of biting prey directly, they may have pinned victims against surfaces above them. This orientation implies hunting along ceilings of bark cavities or under leaves. Modern ants hunt primarily forward, suggesting ecological niches once existed that no longer remain. Another peculiar discovery includes a wasp with an extremely long ovipositor far exceeding body length. The organ may have penetrated deep into wood to reach concealed hosts. While some modern species also have long ovipositors, the extreme proportion in this fossil indicates a host living far beneath bark layers, perhaps a species now extinct. Certain beetles show enlarged eye regions divided into separate upper and lower fields. This design may have allowed simultaneous monitoring of sky and ground, protecting against aerial and terrestrial predators at once. Today, such dual vision is rare, implying that ancient predator communities differed enough to favor this adaptation. Even spiders in amber sometimes display unfamiliar forms. One specimen shows unusually flattened legs extending sideways like spokes of a wheel. This shape suggests the spider hid pressed against bark surfaces, relying on invisibility rather than web building. Later evolutionary success of web building spiders may have reduced survival of these flat ambush hunters. Some of the strangest organisms are microscopic. Tiny arthropods with segmented antennae and multi-jointed mouthparts appear in certain deposits without modern equivalents. Their ecological role remains uncertain. They may have fed on fungal films or decaying organic matter unique to that period's forests. These unusual morphologies reveal that evolution is not a straight line toward modern species. Many body plans were tested and discarded. Amber preserves these lost experiments because small, delicate organisms rarely fossilize elsewhere. Without amber, they would remain unknown, leaving an incomplete picture of biodiversity. Studying these forms also clarifies why modern ecosystems appear stable. Current species represent survivors of long competition. Those preserved only in amber illustrate alternatives that failed when environments shifted or competitors improved. The diversity of extinct designs suggests ancient forests were even more complex than today's. Specialized niches existed for creatures adapted to narrow conditions. As climate, vegetation, and predators changed, many niches disappeared, taking their inhabitants with them. Chapter 7 continues by examining how the disappearance of these strange species helps scientists understand extinction processes and evolutionary selection within ancient ecosystems. When scientists compared strange amber organisms across different geological ages, they noticed a pattern. Many unusual body plans appear only within limited time ranges and then vanish completely. Their disappearance was not always sudden. In some deposits, the number of specimens gradually decreases until the species is no longer found. This pattern suggests extinction driven by environmental pressure rather than a single catastrophic event. One example involves insects with extremely long sensory appendages. Early amber layers contain them frequently, but later layers show shorter antennae species dominating. Longer structures may have been useful in stable, dense forests where detecting distant movement mattered more than speed. As forests changed, perhaps becoming more open or wind-disturbed, shorter and sturdier antennae provided better survival. Natural selection gradually favored compact designs, and the long antennae lineage faded. When all amber discoveries are considered together, they form more than a collection of preserved organisms. They represent a continuous biological record spanning vast stretches of time. Unlike rock layers that often separate species into isolated intervals, amber captures communities repeatedly across different ages. This allows scientists to observe gradual ecological change rather than sudden appearance or disappearance alone. In early deposits dominated by cone-bearing trees, insect diversity centers on bark feeders, sap drinkers, and wood-boring larvae. Predators adapted to these prey types appear frequently. As flowering plants become more common in later deposits, the entire structure shifts. Pollinating insects increase, nectar-feeding behaviors appear, and new predator strategies develop to hunt these faster-moving species. Amber shows this transition directly through changing associations of pollen and insects. The introduction of flowering plants also altered forest structure. Dense clusters of flowers encouraged insects to gather in specific areas rather than spreading evenly across bark surfaces. Predators responded by developing ambush tactics near blossoms. Specimens preserved near flower fragments often show hunting postures positioned toward reproductive plant parts. This indicates that plant evolution reshaped animal behavior across the ecosystem. Climate variations appear repeatedly in the amber record. Warmer intervals correspond with greater species diversity and presence of tropical adapted organisms. Cooler periods show reduced diversity and dominance of more generalist species. Instead of a stable environment, the ancient world experienced continual fluctuation and communities reorganized accordingly. Certain lineages persist across many deposits with minimal change. These organisms likely possessed flexible diets or habitats. Others appear briefly and vanish when conditions altered. The pattern demonstrates that resilience depends on adaptability rather than specialization alone. Amber captures both extremes, side by side. Interactions among species also changed over time. Early parasitic insects were relatively simple, attaching externally to hosts. Later deposits reveal more specialized internal parasites and complex life cycles. This suggests evolutionary arms races where hosts developed defenses and parasites evolved counter-strategies. The preserved evidence allows these biological competitions to be traced through millions of years. Forest disturbances are recorded in multiple forms. Layers containing abundant charcoal particles indicate periodic fires. Immediately following such layers, resin inclusions often contain large numbers of scavenging insects and fungal spores associated with decay. This sequence suggests ecological recovery after destructive events. Amber therefore documents not only stable conditions, but also cycles of damage and renewal. Water availability also influences communities. Deposits with freshwater microorganisms contain many aquatic or semi-aquatic insects, while drier periods preserve mainly bark-dwelling species. By mapping these occurrences across time, researchers reconstructed shifting landscapes from humid wetlands to relatively dry woodlands within the same geographic regions. The long continuity of amber records allows comparison of ecosystems before and after major biological turnovers. Some species persist unchanged through environmental shifts, implying strong adaptability. Others vanish precisely when plant types or climate cross certain thresholds. These correlations help identify which factors drive extinction most strongly. The cumulative evidence portrays prehistoric forests as dynamic networks shaped by climate, vegetation, and biological competition. Rather than a static ancient world, amber reveals continuous adaptation. Each preserved organism is a single moment, but together they form a moving sequence across deep time. This perspective changes the purpose of amber research. It is not merely about describing strange creatures, but about understanding how ecosystems respond to change. The same principles operate today. Species expand, contract, and disappear according to environmental pressure. Chapter 8 concludes by considering how this ancient archive informs our understanding of present biodiversity and the long-term stability of life on Earth. The complete amber record presents a rare opportunity to compare past ecosystems with the modern world. Because amber preserves delicate organisms in precise detail, scientists can evaluate how much life has changed and how much has remained consistent across immense periods of time. The conclusion is both reassuring and cautionary. Many ecological relationships endure for tens of millions of years, yet entire communities can reorganize when environmental conditions shift beyond certain limits. One of the clearest lessons involves stability of interaction rather than stability of species. Pollination, predation, parasitism, and decomposition appear continuously from the oldest deposits to the youngest. The specific organisms performing these roles change, but the functions remain. A pollinating fly may disappear while a bee lineage replaces it, yet flowering plants continue relying on animal transport. A predatory beetle lineage may vanish, while ants take over its ecological niche. Amber shows that ecosystems preserve structure even as participants evolve. This pattern demonstrates resilience at the system level. Life reorganizes instead of ending. However, the record also shows that the transition is not gentle. Many specialized organisms vanish permanently during environmental transitions. The loss is not visible if one only looks at surviving groups, but amber preserves those that did not persist, revealing biodiversity far greater in the past than modern ecosystems alone suggest. Climate change appears repeatedly as a major driver of these reorganizations. Shifts in temperature and humidity alter plant communities first. Because most animals depend on plants, either directly or indirectly, the entire food web follows. In warmer intervals preserved in amber, tropical species expand and diversity increases. During cooler phases, specialized heat-adapted species disappear, replaced by fewer generalists. The change does not eliminate life, but reduces variety. The spread of flowering plants illustrates another long-term transformation. Early forests, dominated by cone-bearing trees, supported particular insect lineages. As flowering plants diversified, nectar feeders and pollinators expanded dramatically. Many older feeding strategies declined. The event did not destroy ecosystems, but rewrote them. Amber records both the disappearance of earlier specialists and the rise of new groups adapted to the new environment. Parasites also reflect stability and change together. Blood-feeding insects existed in ancient forests, much as they do today. Their mouthparts and feeding behaviors changed little. Yet, the hosts they fed on evolved continuously. Parasites persisted by shifting between host species rather than disappearing. This adaptability explains their long survival across geological time. Another lesson concerns recovery after disturbance. Evidence of fire damage followed by bursts of fungal and insect activity demonstrates ecological succession. After destruction, opportunistic species colonize first, then more complex communities rebuild gradually. This pattern occurs repeatedly across different deposits separated by millions of years, showing that recovery is a fundamental property of living systems. However, amber also warns that recovery does not restore the original community. After each disturbance, the rebuilt ecosystem differs from what existed before. Some lineages never return, others appear for the first time. The forest continues, but its composition changes permanently. Over vast timescales, these cumulative changes reshape global biodiversity. The preservation of microscopic organisms reinforces this conclusion. Many bacteria and fungi appear similar to modern relatives, indicating strong functional stability. Yet, numerous insect forms, once common, are absent today. Large-scale ecological roles remain, but fine-scale diversity shifts continually. From a broader perspective, amber serves as a long-term observational record of evolution in action. Each deposit captures a moment, but the sequence across ages shows direction without predicting a fixed outcome. Evolution does not aim toward complexity or simplicity. It follows opportunity and constraint. When conditions favor a trait, it spreads. When conditions change, the same trait may vanish. The ancient forests that produced resin no longer exist in their original form. Continents moved, climates shifted, and species transformed. Yet the principles governing life within those forests continue unchanged. Organisms compete, cooperate, adapt, and sometimes fail. Amber preserves these processes with unmatched clarity because it records not just skeletons, but living interactions. In studying these golden time capsules, scientists do more than describe extinct creatures. They observe the continuity of biological rules across deep time. The details differ, but the patterns repeat. The record demonstrates that life on Earth is both durable and fragile. Durable because it reorganizes endlessly. Fragile because individual species are temporary participants. The strange ancient creatures trapped in amber therefore represent more than curiosity. They are evidence of a world constantly reshaped by environment and adaptation. Each specimen is a reminder that the present ecosystem is one moment in a much longer story, preserved in resin drop by drop across millions of years.